Good morning. As the continuation of the topic simple tissues and their structure and function, in this session, the next simple tissue is coming as the collenchyma. So, these are also the collection of similar cells with a common function. And what are collenchyma? Collenchyma are living cells and the cell walls show localized thickening. As we see in the first type of simple tissue, it is named as parenchyma. The cells do not show any cell wall thickening. But here in the second type of cells or tissues, which is named as collenchyma, which show localized thickening. That is the importance. That is the structural difference from a normal parenchyma to a collenchyma. Naturally, it is originated or the collenchyma are derived as basically the fundamental tissue, that is the parenchyma, but occasionally or due to the specific functions, the cells which show localized thickening on their secondary walls. And it is more present wherever it is seen. It is more present in the petiole of leaves, primary body of stem, that is in the hypodermis. What is primary body of stem? As the shoot development takes place, before secondary thickening, the stem which show the hypodermal differentiation, and in the hypodermis we can see the presence of these collenchyma cells, and it is absent in, particularly absent in roots, but it is present in aerial root. So, normally the collenchyma cells which are present in stem, and especially it is mentioned that it is in the hypodermal region of primary stem, and of course as we see the cross section or in the internal morphology of a root, there we can see the collenchyma cells are absent, and but of course it is present wherever it is in aerial root, those plants which grow an epiphyte and which produce the aerial roots, that is the roots which are growing in the air, which show the presence of the collenchyma cells inside the root system. Okay, and the cells are generally elongated. So, as in the case of parenchyma, it is isodiametric, that means it is somewhat equal in size as in parenchyma. But here in the structure which reveals that the collenchyma cells are generally elongated and with an oblique end walls. The end to end, the cells which show an oblique nature, that is it is in an irregular nature, not only in the similar fashion of structural differentiation. So, cells are generally elongated with the oblique end walls as in collenchyma and are of three types. Here also we can see the three types of collenchyma cells. Here you can see the first one is named as angular collenchyma. And the second one is which is called plate collenchyma, and the third one which is called lacunate collenchyma. Why? Why this such differentiation among the collenchyma cells which are differentiated in stem? Because as the first is named as angular collenchyma, normally the cells which are derived as a fundamental parenchymatous tissue, but due to the differentiation or de-differentiation of cells, accordingly what it is present with what and the localized thickening. What is it is localized? So, here and there or somewhere the cells which show the particular region of the secondary wall show the thickening. So, as in angular collenchyma, we can see the intercellular spaces of the cell, and also the thickening is somewhat extended towards the inner regions. Here you can see in the diagram the intercellular spaces also filled, and in addition to that the corners of the neighboring cells which may also show a little bit of thickening, and which is appears as angular in shape. So, each corners of the cell which show the angular thickening. So, such type of cells which show angular thickening, and they are called angular collenchyma. And the second one plate collenchyma. Why it is called as plate? Because here also the lateral walls which show the thickening. Here any two of the walls, that is the cell wall which show the thickening, and which is called as the thickening at the tangential walls, and such type of thickening which is exhibited in these parenchyma cells or fundamental tissue, and they are called as plate collenchyma. And the third one is lacunate collenchyma. Why it is called lacunate? Lacuna means the gap. So each cells differentiated as a fundamental tissue, and thereafter the localized thickening which has happened, and that is why those type of cells are called as collenchyma. And here the third type, the thickening at intercellular spaces alone, that is each cells which show only the localized thickening inside the intercellular spaces, while we can see the differentiation as in the case of angular collenchyma, not only the intercellular spaces, but in addition to that the inner walls of the each corners which show triangular thickening. But as in lacunate collenchyma, only the cells which show the lacuna is getting filled, or the intercellular spaces become thickened, and such type of cells are called lacunate collenchyma. So here the collenchyma is getting differentiated into three types. One is angular collenchyma, second is the plate collenchyma, and third one is lacunate collenchyma. Okay, got it. So we can review it. What is the collenchyma? Collenchyma are living cells. They show localized thickening, and which is commonly present in petiole of leaves, primary body of stem, that is in the hypodermal region, and especially the collenchyma cells are absent in roots. Only such collenchyma cells are present wherever it is in the aerial root, and they are generally with what? Generally it is elongated, and the end walls or the walls become oblique in nature, and they are of three types, which is called as angular collenchyma, plate collenchyma, and lacunate collenchyma. It is corresponding to their thickening. Okay, thank you.